The study of epigenetics holds very close ties with brain development. In order to fully understand the way a brain develops, it is important to understand the epigenetic mechanisms that drive it. This summer, I had the opportunity to work with the Miranda Lab and closely study a popular model for this concept, axon pruning in the fly brain. To give some background, axon pruning is a process that involves the elimination of improper neural connections within the brain, hence the name axon pruning, as it is pruning away the connections which are called axons. How does epigenetics play into this? Well, epigenetics studies the changes in the way a gene is expressed. The process of axon pruning is regulated by these changes. While extensive research has already been done on axon pruning in fly brains, there remains a substantially large gap of knowledge in the epigenetic mechanisms that drive it. To shed light on this, we focused on an epigenetic relationship between two fly proteins, Kismet and HDACs. Their interactions can be compared to that of a seesaw. When Kismet is defective, we experience an epigenetic imbalance that results in axon pruning defects. However, when HDACs are knocked out via HDAC inhibition, this balance is restored, preventing such defects. Now, our lab already determined this interaction, but the issue that remains is that there are 5 kinds of HDACs in flies, and we have yet to identify which one is responsible for this balance with Kismet and axon pruning. So that brings us to my current study, where we identified HDAC1 as a strong candidate. Now, how did we get here? Well, we mated flies using a biochemical technique known as the Gal4 UAS system, which essentially allowed us to turn off either the Kismet gene and HDAC gene. Since turning off the gene renders that protein absent, it allows us to experiment with the Kismet HDAC balance. We then dissected the offspring brains and used confocal microscopy to capture images of them. Using the images, I measured and compared the surface area of the axons in the brains. To give you an idea of this process, here's what an axon pruning defect looks like. You can see the axons here. Now this is what proper axon pruning looks like. As you can see, this axon is much smaller than the one on the left. Now, we expected that eliminating HDAC1 would prevent defective axon pruning. Interestingly enough, we found the complete opposite. As you can see here, eliminating HDAC1 resulted with a considerably larger axon in comparison to the control. This suggests that contrary to our expectations, loss of HDAC1 in particular may actually cause defective axon pruning rather than fix it. This also suggests that there needs to be a sweet spot in eliminating a combination of HDACs for a healthy axon pruning. Sadly, due to the limitations imposed by the current pandemic, HDAC1 was also the only HDAC that we were able to test, which means we can't 100% confirm that these results aren't simply due to error. But it does provide us with a new lead in testing HDAC and Kismet interactions. And on a broader view, this lead also provides us with a potential target in treating brain disorders that are associated with this epigenetic relationship. Ultimately, this research experience was fruitful and rewarding, both in how I developed skills as a researcher and contributed valuable knowledge to the research community.